Over the last half-century, geneticists have made sense out of what was once a nebulous universe of molecular matter. But there is always more than meets the microscope. In recent years, a few scientists shook the genetics community by shining a light on the dark matter of cells. Victor Ambros grew up on a small farm where his love of science grew out of a passion for astronomy.
I lived in Vermont, so the skies were dark, and my father and I would go out there and sort of just talk about the stars and the wonders of the universe.
As a boy in California, Gary Ruvkun was also infatuated with space science in the 1960s.
There I would, a little nerdy 8-year-old waking up at 3 or 4 in the morning, watching the launch. It was 1,000 times better than anything at my school.
Both ended up focusing their research not on the vast reaches of space, but on a smaller universe, the intricate world inside a living cell. The kindred scientists would meet in Boston while earning their PhDs. Later, while running their own labs, they stumbled upon a surprising connection between their lines of research. While Ruvkun's Harvard team was studying one gene of the microscopic worm C. elegans, Ambros's MIT lab discovered that a nearby gene produced a curiously small strand of RNA.
And so Victor and I together in June 1992 had a phone conversation at about midnight, comparing his sequence and our sequence, and it turned out the important parts matched like a hand in a glove.
To their amazement, the small molecule of RNA coded by the one gene was somehow affecting expression of the other.
So the idea that such a small molecule could be a regulator was really quite astonishing.
But it would take the work of another scientist across the Atlantic Ocean to indicate just how significant these small RNAs really were. Since the mid-1990s, David Balcom had been researching the fascinating process of gene silencing by studying viruses in plants.
And indeed, we found that the genetically modified plants were resistant against the viruses, as we expected. But we found that in the plants that were resistant, the newly introduced viral gene was actually switched off. So then we started asking why.
It wasn't until 1999 that he discovered the silencing was associated with RNAs amazingly similar in size to those identified by Ambros and Ruvkun.
The only place we had not looked was in the part of the analysis where we would have detected very small RNA molecules. So then we started looking at those and straight away found them.
So that convergence told us then that these small RNAs were part of a suite of phenomena that must have evolved prior to the separation of plants from animals.
They're living fossils, they're relics that still exist in our genomes.
A unified principle of small RNAs and their role in gene regulation quickly surfaced. Double-stranded RNA is chopped into different types of small RNAs, which guide enzymes to turn off other genes.
Of course, expression of genes is not just a question of switching things on at the right time, it's also a question of switching things off at the right time.
Thanks to this groundbreaking work, a completely new class of regulatory molecules has been uncovered. The tiny molecular bits, once thought to be cellular junk, are actually powerful control switches affecting all kinds of life processes. The silencing ability of small RNA could revolutionize everything from the engineering of crops to the treatment of cancer.
We could use silencing-based technologies, for example, to switch off genes that cause fruits to rot.
In C. elegans, we can routinely knock down genes by simply feeding the worm bacteria that's producing a double-stranded RNA.
We showed that we could cure effectively the cancer that a worm gets by inactivating RNAi. And so it is quite a rage now in oncology, and it's extremely satisfying.
If we can get the small RNA into the cell, it'll do the work. So I'm really that optimistic.
The 2008 Benjamin Franklin Medal in Life Science is presented to Victor Ambros, Gary Ruvkun, and David Balcom for the discovery of small RNAs that turn off genes. Their pioneering work initiated a paradigm shift in our perception of the way genes are regulated. And this insight is making possible major new genetic tools for basic research and for improving agriculture and human health.